Thank you. Yes, wonderful. So yeah, while I'm pulling up my slides, hi, my name is Anna. I'm a postdoc in the Grutter Lab. I actually got my PhD at Iowa in applied mathematics, but then Chad recruited me to help him characterize a MED13L heterozygous or het mouse model, and that's what I'm excited to talk to you all about today. Okay, so as Matt mentioned, mouse models are a great scientific tool because we have such a high similarity between the human genome and the mouse genome. Let's see if I can put my cursor on this. Oh yeah, great. So the laser pointer, it'll just be a cursor and that'll be fine. So comparing human MED13L gene with a mouse MED13L gene, we can see that it has about a 93% similarity or percent identity, which is also very similar to a rat MED13L gene, which is right around, so I think this one is around 93, 92, and then compared to a zebrafish, which is just over 50% similarity. And so then if we look at, let's see if it pops up. Okay, there we go. And then, so if we compare, again, human MED13L protein structure, so this is the 4 different domains that make up MED13L protein. If we compare the human with the mouse, again, we have very high overlap. And for the particular mouse model that I'll be talking to you about today, we have deleted exon 11 from MED13L, and exon 11 is in this disordered region. And so, as Kayla mentioned a little bit earlier, how we go about deleting this exon is we first insert something called a LOXP site into our genome, which we can then activate through something called a CRE system. And so the CRE activates these LOXP sites, which then essentially cut out the desired exon. And so our mouse models today have exon 11 deleted from them. And we discovered in breeding these mice that a homozygous knockout is embryonic lethal. So if both copies of MED13L do not have exon 11, then it's not viable. It doesn't make it to a post-embryonic stage. And so how we discovered this is thinking about your Mendelian ratios. So you would expect 25% of the mice born to be wild type, or mice that have 2 non-mutated copies of MED13L. You'd expect 50% to be heterozygous, so 1 copy with exon 11 deleted, 1 normal copy. And then you would expect 25% to be that homozygous knockout, which we do not see in our mice. We have 64% heterozygous and 36% wild type. So our homozygous knockout is not viable, which is a little bit different from what Matt was seeing in some of his mutations. So moving on. So what I'm going to show you today is some growth data from our heterozygous and wild type mice. We'll also look at some cardiac data, cranial facial data, and then we'll finish it up with just a few behavioral assays. So starting first with our growth data, we'll look at the females first. So you have your het females in pink and your wild type females in black. And you can see, well, I guess first I should say, these mice were weighed once a week for the first 8 weeks of life. And the het females are statistically smaller than their wild type counterparts. But if you look at the sort of growth rate from week to week between the average weights, you can see that even though they're smaller, they're still growing at very similar rates and following that same growth pattern. So at least in our mice, we believe that they're smaller at week 8 because they started smaller and then they're just never able to make up that initial sort of difference. And then similar in males, but less obvious, so still statistically significantly smaller at week 1 and week 7, but as you can see, there's a little bit more variability from week 1 to week 8, and again, The het males follow the same growth curves as their wild-type counterparts, but they're just sort of starting out slightly smaller and then maintaining that difference, but again, not as obvious as the females. But so we see that our MED13L het mice do experience a growth deficiency. And so then moving on to some echocardiograph data. So we wanted to see how aging affected the cardiac function of our mice. And so we had the mice undergo an echocardiogram at 4 months and 12 months. And you can see that in terms of heart rate and ejection fraction, which you can think of as the efficiency of the heart, we're not seeing any differences at this time. So our MED13L het mice do not exhibit cardiac abnormalities as of 12 months old. These mice actually turn 18 months next Tuesday, and so I'll be doing more echocardiograms next Tuesday. So these mice are currently still alive, and still we're just sort of watching them as they age naturally. There's no stress involved in this sort of experimental setup, just natural aging. So then moving into the cranial facial data. So we'll start with a picture of a micro CT scan of a wild type mouse. And so just to orient ourselves, this is sort of the front of the skull. You have the nasal bone, and then this is a bottom, sort of looking from the bottom up. So you have bottom jaw, bottom teeth, and then the upper teeth. And so this is a wild type mouse. This is a heterozygous mouse, and you can see it looks very similar to the wild type mouse. This is another heterozygous mouse, and specifically in this photo, you can see that the nose is stunted, or it's more pug-shaped. And then this is another heterozygous mouse, and here we have sort of a crooked nose phenotype. So we observed 2 midfacial anomalies in our MED13L het mice. We call this our pug phenotype, and then this is our sort of crooked nose phenotype. And out of the 23 mice that we categorized in this way, we found about 39% of our het mice had this normal snout that was very similar to the wild type. About 21% had like the pug snout, and then about 39% had the crooked snout. And so in terms of the pug snout and the crooked snout, those types of phenotypes are sometimes caused by craniosynostosis, which is when your suture lines and your skulls fuse together. So in a standard suture, there's a little bit of a gap, but if the sutures fuse, you lose that gap, and then you might have some of these phenotypes because of that. And so we investigated this by staining our skulls. And so we wanted to compare the lambdoid suture, the sagittal suture, the coronal suture, and then also the mectopic suture. And you can see that we didn't actually observe any craniosynostosis in terms of any of those sutures that I just listed. You have your wild type mouse and then your het mouse. But what we did see is a discontinuation of the squamosal suture, so that's on the side of the mouse skull or the human skull as well. And so in a subset of our hets, so 3 of the 6 that we photographed had this discontinuation. And so you can see right here the dotted yellow line has disappeared, and that's because in this particular area the squamosal suture has fused together. And this discontinuation was irrespective of side of the head. So here, this is the left side of the head, this is the right side of the head, and so it's only discontinuous on one side, whereas the other side is normal. And so this is just the wild type that you can compare to. And we found this very interesting because, as I'm sure you're all aware, there's been a couple cases of MED13L patients that have also had craniosynostosis. I believe those cases, though, were in the mectopic and coronal. So this is just another suture that we're seeing fused. Okay. So then I'll move into some of the behavioral assays that we did. So we'll start with something called an open field test, which Matt briefly discussed. And so the open field test measures anxiety in our mice. And so the first thing we looked at was total distance traveled during the extension of the test. And we saw what maybe is a slight decline in the movement of our het females, but it wasn't statistically significant, and you can see there's a lot of variability here. But we did see a statistical difference in the time spent in the center versus the time along the outer edge. So our wild type females in black spent more time sort of running around the entire maze, spent more time in the center compared to their heterozygous counterparts, which spent more time sort of along the outer edge. And so this could indicate that there's maybe higher anxiety in our females, but we did not see that difference in our male mice. Something else that we did is a Barnes maze, which is looking at learning and working memory. And so the idea behind this assay is that each mouse is assigned an escape hole. And so then over the course of 5 days, they learn where their escape hole is. And so we can see that on day 1, the het females traveled further to find their escape hole, but then after day 1, they learned where their escape hole was, and then they also seemed to remember where their escape hole was from day to day. We did not see the same difference in the males on day 1. They seemed pretty similar throughout the entire test, but something that we did observe in both sexes was a difference in how they entered the hole. So this is a wild type male on day 4. He finds his hole and he goes in, compared to a het male on day 4. And so you can see there's a little bit of a stutter or a hesitation, possibly due to increased anxiety, possibly due to something going on with depth perception. We're not actually sure. Unfortunately, the Barnes maze was not the correct assay to sort of quantify that behavior. It was just something we observed in watching the mice as we trained them for 5 days. So this is definitely in our future goals to go back and quantify and try to investigate what could be causing that hesitation. Oops. Okay, and so lastly, we put the mice on Rotarod, which is for testing motor ability or motor learning. And so we'll again start with the females, and we can see that on day 5, the het females traveled distance traveled. Okay, so on day 5, the het females traveled less than their wild type counterparts. So you can see they sort of increased for the first few days, but then they plateaued a little faster than their wild type counterparts, perhaps due to decreased motor learning. We did not see that same difference in the het males. There was almost too much variability. There were these 2 guys that were just really good at it. But we did see our wild type males fell off a lot on day 1, and we think that was just because they were a little heavier than their het counterparts. And so in conclusion, our MED13 het mice exhibit growth deficiency, 2 types of midfacial anomalies, possibly increased anxiety, and then possibly a learning disability in terms of motor learning. And so where are we going next? We're going to wrap up some more behavioral studies, as I mentioned, but we're then going to transition into an MTMG mouse, which is going to allow us to isolate our mutation to the neural crest. And so then we can start to tease apart a mechanistic understanding of MED13L's function in like neural crest and early development, because right now the data that I showed you today was a full body knockout, and it's just really hard to get into like the mechanistic approach when you've got the mutation in the entire body. So we're gonna try to pinpoint it a little bit. And then, as Kayla also mentioned earlier, we're going to be doing some collaborative work with Dr. Brad Gruder, Chad's brother, to investigate neuronal activity with some electrophysiology. And with that, I'd like to give a special thanks to the MED13L Foundation for putting on this event and for giving us the money to do this. I really enjoyed this project, and I know my lab mate has also enjoyed working and meeting you all. And then another special thank you to the Van Otterloo Lab at Iowa, who helped us photograph our skulls and get that micro CT scan data. So thank you.